Right, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon. I'm not gonna wait for anyone, so we will just continue. Oh God. Okay. So what we will do today is we will start to look at. Never mind. I'll just go per slide then. This is just for those who actually have not completed post intervention B. Okay, intervention B is the pre-recorded thingy whereby you go and answer all these surveys or not survey tutorials in during your lecture viewing. Okay, so if you have not done so, do help me in completing the survey. Okay, all right, and this is for today's attendance. I actually forgotten to prepare one slide for the attendance, so I'll just do a screenshot from my other computer. But I've tested the QR, it should work. So you can just quickly go grab your phone, scan this, and key in your code, then you're done. All right. I'll give you guys 5 more seconds. All right, 5 seconds. Okay, so at the beginning of week 8, if I recall correctly, okay, I actually showed you guys this slide whereby what are the things that you'll be learning is basically proteins, which are two categories. One is proteins and peptides, the other one is nucleic acid. So under the sub heading of proteins, you will learn about amino acids, peptides, proteins in terms of the structure, how you can actually synthesize them, and how you can actually analyze them. Okay, so on Tuesday we actually did this. Okay, we actually looked at how we can actually degrade the amino acid and thus be able to analyze the sequence. Okay, so I didn't have any summary for today and any poll F because if I do have them, I don't think I can finish the intended lecture during the time slot. So I skip them altogether. If I manage to cover whatever I want to cover next week on Tuesday, then we will have a short poll F questionnaires or tutorial, and then on Thursday, we will have your second quiz and the last class for the whole semester. Okay, right. So moving back to the lecture content. So proteins, on Tuesday, we've looked at the degradation analysis. We've looked at full hydrolysis. Okay, for example, using 6 molar of hydrochloric acid, 100 degrees temperature, and then give it about 24 hours. Then you manage to, you'll be able to hydrolyze the whole structure of a protein or peptide into its individual amino acid component. Okay, we also look at half, how do I spell it? Half hydrolysis or partial hydrolysis or whatever the terminology is. I don't mind. It's not like a name of a reaction. It's just pretty much half of that one. Okay, meaning that you did a little bit of hydrolysis but not a total hydrolysis. Okay, we've looked at a few ways by which we can play around, okay, and do it, and either via chemical or biochemical approach, okay, enzymatic or full chemical. And finally, we've looked at the Edmond degradation technology. Okay, Edmond degradation, by which you actually digest out one amino acid at a time from the N terminus. And we also looked at using enzymes to help you to digest one amino acid at a time from the C terminus. Okay, and the comparison between those two technologies, of course, Edmond degradation is better because Edmond degradation is independent of time, while the C terminal hydrolysis, it depends because the enzyme that is being used are not specialized or specific towards C-terminal degradation. So depending on which amino acid that you want to digest, sometimes it might take a while, sometimes it might take a short time. So Edmond degradation is the better option for sequence analysis. Okay, so we have finished all of those. What we're going to look at today is basically nucleic acids. So we will look at what is DNA, what is RNA, but of course, this is not a biological class, so I'm not going to go dive into very detail about each and every single one of these. So we just looked at this basic structure of DNAs, RNAs, different types of RNAs, and the things that are related to DNA, RNA, and chemistry. Okay, so those things will be the main focus. And but something which is more biology is we will look at on how protein biosynthesis works. Okay, and this hopefully by today's class, at least you can appreciate the technology that is being used in Pfizer vaccine. Okay, all right. So this is just for today. For next week, there are a few things that I do have in mind to actually teach you guys or share some knowledge that I have. But unfortunately, there are so many things to talk about in the genetic technology. So I will just touch a few things, especially for PCR. PCR, because I'm sure at least by now everybody have heard the word about QT-PCR, okay, whereby when you get the nose swab or your throat swab, the sample will be sent to IMR or any institutes that have a PCR machine, and then the PCR machine will amplify the mRNA or RNA, the viral RNA that is on your throat or your nose swab, and see if the quantity is high or low. If the quantity is high, meaning that you are a positive swab COVID-19 case. Okay, this is just an example. I'm not saying that PCR can only be used for COVID-19. No, it's amplification of those two types of molecules, DNA and RNAs. Okay, but one of the other application applied use of PCR is to quantify the number of viral mRNA. Okay, so that's what we call, if you read in the newspaper, we call it as QT-PCR. So we will look at that on Tuesday next week. Okay, so we will also look at a little bit on the principles of biotechnology or genetic technology, and how you can actually utilize all these technology in forensic DNA analysis. Okay, throughout the lecture, you will still hear me coughing. So I'm hoping that you do not have to go full blast on your speaker, because otherwise, if sometimes I accidentally cough very, very strongly, I hope that will not damage your eardrum. Okay, all right, so we are already on, oh, sorry, that one. Okay, so we passed through that one last Tuesday, and now we are on the 10th of June. So we have one more lecture on nucleic acid next Tuesday, and finally we will have your quiz. And your quiz, of course, will only cover this section. Okay, so I will not be putting whatever we have covered in that particular lecture over here, but for your final exam later on, this two, well, pretty much it covers the whole subject, so definitely will be there. Okay. Now, probably I'll just talk about you. Yes.
My friend are waiting to be accepted.
Oh, I thought it should be automatic. Sekejap lah. Sekejap ya. I actually set an automatic acceptance on my PC. Fortunately, it didn't work. Let's see why it didn't work. View admin all. Okay, done. Okay. Background noise, guys. Don't worry about it. I think everybody should be in by now. Okay. All right. So you can see the. Yep. Now, right. So now the auto admit session is back online. All right. So thank you for letting me know. Okay. So nucleic acid and introduction. So what are nucleic acids? So I put the title as Nucleic Acid, a love story, because I found this kind of like caricature between a DNA and an RNA, and the DNA says still single and RNA says always. Okay, um, it's funny because this only applies for a very old technology. Are people still waiting, or they managed to come in? Okay, they managed to come in. Okay, so it's funny because um, nowadays RNA can be a double-stranded RNA as well. So this is a very old thing, and I find it funny because people are still using this, and the technology have advanced, moved past through this, but whatever. Okay, so we've looked at the central dogma of molecular biology, whereby DNA produces RNA, and then RNA produces protein. Okay, so that is the basic. But I also mentioned that RNA can also transform into DNA. Okay, biologically it's possible. Of course, chemically it's always possible. You can always do whatever you want. But in terms of biology, how it works is there are some viruses, especially that regenerates DNA from an RNA. Okay, but unfortunately, COVID is not one of them. So the only virus that I can recall now on top of my head is HIV. So human immunodeficiency virus, or the ones that cause AIDS. Okay, so that virus is very dangerous, and it likes to convert things from well, it doesn't like to do a normal way, so it will just do whatever it wants. So in this case, originally it's a RNA, and then it converts its own RNA to become a DNA. Include your DNA into your cell, and then your cell will produce mRNA, and then from mRNA it will produce multiple copies of itself. Okay, so it's a very dangerous virus. All right. So just based on this central dogma of molecular biology, we know that well, it's kind of like a love story. DNA is always going after RNA. At least that's what we want. Okay, but some of you might heard about the word DNA, RNA, and we've looked at what proteins are. So now we want to look at what is RNA and DNA. Okay, so basically, DNA is our genetic material. Okay, it's kind of like a memory about us, but it's not a memory that you, in a sense that you remember what happened in the past. Okay? It's more on what happened to us as a human, and throughout multiples or millions of generations. Okay, so this is what DNA molecules are. Okay, so it encodes in a polymeric molecule called a nucleic acid. So that is the general term about nucleic acid. So nucleic acids are generally divided into two categories. Of course, there are multiple more subcategories that we will look at in the next few slides. But in general, there are two categories. One is called as deoxyribonucleic acid, and the second one is ribonucleic acid. So as a chemist, by just pronouncing the name, you should know that deoxy means no oxy. Okay, no oxygen. As simple as that. Okay, ribo is a type of sugar. So I hope that you at least encountered something about ribose sugar during your class with Dr. Dini. Okay, and nucleic acid, you know that is an acid. As simple as that. So by just the naming, if you compare between a DNA and an RNA, you should know that, well, DNA doesn't have, well, it has the same nucleic acid or ribonucleic acid, but RNA has extra O. DNA does not have extra O. So if you look at the structure on the right hand side, oops, okay. So if you look at the structure on the right hand side, ignoring the base, so this is the base component. So we look at the components after a while. We're just focusing on just the naming. Okay, so we have ribonucleic acid. One is deoxy, one is just ribonuclease. So if you look at this one has an extra O compared to that one. Everything else is almost identical. So you know that this one is an RNA molecule while over here, because it doesn't have any oxygen, it's the deoxy um, ribonucleic, D and A, ribonucleic acid. Okay, so that is the key determining differences between RNA and a DNA. Okay, of course, function wise and so on and so forth can be differ, but we're focusing on the chemical structure differences between DNA and RNA, and these are the this is the main difference between an RNA and a DNA. Okay, and of course, DNA function to carry genetic material in almost all living organisms. So I put there living, okay, because regardless of who you are, whether you are a squid in the ocean or a bird in the sky, or simply just a cheetah running around in the field in Africa, everybody has the same DNA, as in the same molecule of DNA inside your body. Okay, so but if you consider bacteria, on the other hand, there are some unique bacteria that have slightly different DNA molecule, but most of us, I would say 99% of all living being in the world, at least in on Earth, okay, we have the same genetic material. Okay, now ribonucleic acid, on the other hand, not it's still the same. It's still a genetic material, but I call it intermediary material of all organism. So this one is including viruses. Okay. So why do I categorize them into two and did not put viruses under living organism? It's all because of a definition of living. According to let me recall, a biological definition of a living being is an organism that is able to replicate on its own. Okay, so a bacteria, they can replicate on its own, but viruses like COVID-19, they cannot replicate on its own. Okay, it needs to have a host. Therefore, COVID-19 infects human becomes the host, and then the host helps the viruses to actually replicate and infect other people. Okay, so viruses are one of the ambiguous category. You don't call it as living, but you neither call it as dead, because it can replicate, but it cannot replicate on its own. Okay, so viruses belong. Oh, well, again, this is most viruses. There are viruses who contains DNA as their genetic material, but most viruses that at least infect humans contains RNA as their genetic material. Okay, now I've already answered about that, so you don't need to have a educated guess. So what we will see now, you can see on the right hand side where I put in a very small alphabet inside there. Okay, so basically there are five different molecules. So, but all of them, so all of these are connected to either one of these. Okay, so DNA on the left hand side and RNA on the right hand side. Now, what are the main differences? Alanine, guanine, cytosine, and tyrosine are all bases found in, instead of RNA, it's in the DNA. Okay, so these are all the bases that makes up all of us. So as long as you are human from Earth, then you definitely have these four bases. Okay, while for RNAs, so instead of having a threonine, what we have is a uracil. Okay, so uracil substitutes threonine as the molecule during RNA genetic. Oh, how I say this? During the process of converting between DNA to RNA, so our RNA copies contains a uracil instead of a T. Okay, if you look at the structure, they are almost identical, but there's only missing of one methyl group as compared to a threonine. Okay, and if you look at the structure over here, it's a bit ugly because I already drawn my pen with my pencil. But what you'll see here is I highlighted the differences between the bases on the genetic materials. Okay, right. Now, these are some examples of different types of RNAs in the body. Okay, what we've heard normally, especially for Pfizer vaccine, for example, we heard that Pfizer and Moderna vaccine uses mRNA technology. So mRNA this, mRNA that, and then you might have heard about some people say, well, the mRNA will get into our DNA and change us from being, I don't know, non-human perhaps. Okay, so you might have heard about that, but that's why learning about this very critical so that you are well informed about what are the real situation. Okay, so RNA has multiple types. As you can see here, mRNA is just one of the subtypes of RNA. Okay, so what does mRNA do? mRNA encodes, it's a digital code for protein. Even though it's no longer 100% true, but previously mRNA just encodes for protein. So, but nowadays we know that mRNA carries a lot more information, not just about proteins, it also carries about non-protein structure. mRNA can also be transformed into something else. Okay, so it's just a matter of where they go towards the end of the day in the cell that makes an RNA becomes either M or T or R or SN and so on and so forth. Okay, so one example that we um, normally heard day after day is mRNA. We also have in our body a tRNA. Okay, so the T over here means transfer, so transfer RNA. So it helps, so it's an adapter between mRNA and amino acid. So it actually helps our body to carry the amino acid. So well, I actually have a picture, then we will go through the picture and I will talk about it in soon instead of drawing it here. So other than that, we have rRNA, which is a ribosomal RNA. So our ribosome contains, so ribosome, if you have heard, I mentioned about it previously, function as to synthesize proteins in our cell, like 1,000 amino acid per second. Okay, so that's why I mentioned previously. So if it's a protein, then it contains amino acid. But in reality, ribosome is not just a pure amino acid. It also contains pieces of RNAs, and these pieces of RNAs actually helps the whole protein to identify RNAs or mRNAs in a very quick succession. At the same time, identifies tRNAs that carries amino acid. Okay, so we looked at the structure in a little bit more detail in the next few slides. So other than that, you have snRNA, splicing RNA, so SNO-RNAs, mRNAs. So even some of them are quite recent that while I was doing my degree about, let's see, 10 years ago or so, okay, I have not learned anything about this. So snRNA and SNO-RNA, I have never heard about it. So even these two are very new to me, and very recently, as recent as last month, okay, another type of RNA was actually discovered on the surface of our cell. This is very new, and it opens up a lot of applied technologies that can be utilized if we actually know what these RNAs function as. Okay, so maybe in the future, you guys will be the ones who will be investigating about using RNAs for drugs and whatever else. Okay. All right. So other than that, we have miRNA, which is a microRNA. It regulates gene expression. So you know that our DNA contains what we call as gene or genetic materials. So this gene encodes for a feature, okay, in our body. For example, a gene can be as simple as whether your hair will be a straight hair or a curly hair, whether you have a long nose or Asian short nose, whether your eyes are big and wide or small and wide. Okay, so those things are being carried by our genetic materials, and we cannot do anything about it, except for, well, plastic surgery, but it's a different story. Okay, so in order for some genetics, so we know that we are all humans. So basically, we should have similar genetic codes, right? So but we all know that, I'm sure everybody is adults now. Okay, your pubic hair, for example, when you are a kid, so there is no pubic hair. But when you are an adult, you will grow a mustache, different types of pubic hair, and so on and so forth. So what makes you different compared? Well, what makes you different when you are younger and you are an adult now? Okay, so basically your genetic material did not change. So what does change? So one of the control mechanism in our body is this guy over here. Okay, so it regulates gene expression, meaning that it will turn the genetic code in your cell either to turn it on or turn it off. Okay, similarly, if say you are injured, okay, you are bleeding, so the bleed sends signals to your cell to turn on a certain types of gene, so that your body will go into a healing process. Okay, so those are the things and that involve in our body and part of the control mechanism. I'm not saying that mRNA is the only control mechanism, but part of the control mechanism is this. Okay, sRNA, on the other hand, silences gene expression. So these are mainly not in human, it's mainly in invertebrates. For example, in worms or in some type of fish, okay, or even plants use sRNA technology as to silence gene expression. So what does silence mean? It means that it stops the gene from working in the body. Okay, say for example, if this is a tree, apologize for the ugly drawing. Okay, this is a tree, and I'm hoping that some of you guys know that tree can be infected as well. So some of the infection is caused by a specific virus that infects, say for example, the leaf of a tree. Okay, but tree doesn't have antibody like us human. So in our body, if we are being infected by viruses or bacteria, our body fights back by there are multiple ways, but one of the ways is by producing antibodies. So plants, on the other hand, do not have the genetic codes to actually code for production of antibodies. But what they do have is they have a mechanism to turn on the genetic code for sRNA, and this sRNA will then silence the gene of the viruses so that they can heal themselves from the viruses. Okay, so it's very interesting if you go into very detail, because sRNA is like an antibody in our body. It's just it's a different molecule. It acts in a different mechanism, but the function is the same. It is to protect the host from outside infection. Okay, and the last one is RNC, RNA. Again, it regulates gene expression in some other ways, which I need to recall what they are. Otherwise, I don't remember. Okay. And these are another example, some other examples that you can see as RNA. So the ones that I did not learn, it says participate in slicing and transfer of HNRNA. Now a different terminology exists. So there are multiple types of mRNA, RNAs. So they are very, very tiny, but nonetheless, they are very, very critical in various roles and functions as shown in this slide. Okay, so let's move back to look at the general aspect of DNA and RNA and how you can relate this back to chemistry. Okay, so I like this image very much. It was produced by this group, I guess, or app developer called Baiju. Okay, it sums up the differences between these two nucleic acid. So it's shown here. You have a cytosine, that's guanine, and of course, previously I just put in the first letter, okay, like that. So this time around, you can actually see how they are actually spelled out. It doesn't mean that you have to memorize, no, but it's good to just know how to actually spell that. And then you can always see here again, uracil are for RNA only, while thymine is for DNA. Okay, and the uniqueness between these two are they are complementary to one another. Okay, C or cytosine always pair with guanine, and A alanine pairs with either thymine in DNA molecule, or it pairs with uracil in RNA molecule. Okay, so for example, we know that in our body we have DNAs. Okay, and we know that mRNA carry the information for protein synthesis. So how do you actually copy from here into an mRNA, okay, with only a single copy? And then how does this actually transfers into a protein? Okay, so we will look at this in a short while. I think it's next slide, or probably the slide after this. Okay, but we will go into that very soon. Oh, we don't have enough time. All right, so what are the basic structure of a nucleic acid? So a nucleic acid basic structure is as shown here. Again, this is just kind of like a summary of what I've said initially, but it will give like a big fine detail on the structure of the amino acid, nucleic acid. Okay, so it has a 5 carbon sugar, or in the chemistry term is a beta D ribose. So again, you need to recall what are the differences between L and D sugars. Okay, so basically what you've learned from Dr. Dini's lecture can still be applied in our nucleic acid lecture. Okay, and you also need to remember what does a beta and alpha sugar means. Okay, so beta and alpha, I just leave it to you guys to actually recall what those are. Okay, it doesn't mean that I don't know. I do know. I just don't want to tell you guys. Okay, so the 5 carbon sugar are linked to 1 phosphate and 1 base, and of course the base are either this and U. Phosphate is at the 5th position. So what do I mean by this? We will see in the next few slides. But just remember 5th position. Okay, and at the same time, you need to understand why does the phosphate become 5th. Okay, so in chemistry you have what the first carbon. Well, I'm not sure whether you guys learned about this in the sugar section, but I'm just gonna repeat it if you guys did learn about it. Otherwise, just accept it as a new thing. So the first carbon is over there from the base. The second one is over here. Either you have a deoxy or a oxyribose. The third position is over there. So the 4th carbon position over here, and the 5th position is over that one. So when you say it's the 5th position, it means that the 5th position on the carbon is where the phosphate is. Okay, so differences between DNA and RNA is thymine and uracil, as I mentioned, and deoxy versus oxyhydro at the 2 position. Now, so this is why you need to remember what is 5, what is 3. So 5N and 3N. Similarly, or well, slightly different than when we are talking about amino acid, you have the C terminal and then you have the N terminal. So on ribonucleic acid molecule, you always have a 3N and a 5N. So the full name, we will probably, if you actually go into detail, you will hear people saying a 3' N and a 5' N. Okay, and the differences between these two is in biology, the synthesis starts from a 5' N towards a 3' N. Okay, so this is how when we are copying our DNA from a DNA to RNA, it always starts from a 3N, 5' N, moving to a 3' N. So even if you turn the amino acid or the RNA around, so you will have your 3N here, but then you will know that from your 3' N, you have GGG, G, C, 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 blah, 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 and then finally you have A, and then 5'. Okay, so you always know the direction on where the nucleic acid go. Okay, so this is why it's very critical for you to know about this. Now, if you go back and relate to how DNA molecule are, I might ask you in the exam to say, for example, ask you guys write down the pair code of a DNA molecule from the given sequence. Okay, and then if this given sequence like this, then of course what you need to write is actually something like that. Okay, G G G G and C C C C, and then 5 prime. Okay, so this is how we write it. So for amino acid and peptides, you always consider one thing. You just consider where is the C terminal, where is the N terminal. But for DNA and RNA molecules, depending on the situation, you might have two sequences like this, but then one sequence will have a 3' end, the other sequence will have a 5' end. Okay, and why does this matter? Again, this matter because of this. So the 5' end position always have a phosphate. So if you accidentally put down here 3' end, and this one is 5' end, okay, now it becomes something else altogether. The structure of the nucleic acid now differs. Okay, so that's why it's very critical to actually write it in a proper way. So a 3' pairs with a 5', the sequence pair according to as I mentioned. Okay, so guanine always pair with cytosine, and thymine always pair with adenine. Okay, so the pair is always complementary. If it's A, then becomes T. If it's T, it becomes A, and so on and so forth. So this is the basic for the the helix of DNA. Okay, so that's when you will see this type of structure a lot, and say if that one is 3', that one will be 5', and if you follow through, that one will be 3', that one will be 5'. It's always like that, right? Now, but DNA is double-stranded, double helix. You ask, so it's easy. As I mentioned, I've already write it down there. So this is the pair. Okay, so you have a 5' like that. So the 3', the opposite, the pair is the opposite. Okay, of course this structure is a bit ugly because I don't have the font is not similar, so it doesn't really go in a straight line like that. Okay, but make sure that you understand it should be a complete pair. Okay, so it's connected by the strongest bond in biology, which is, I'm hoping that you guys are saying this while keeping your mic muted. Okay, hydrogen bond. Okay, so it's like that. So when you have a thymine and an adenine, you will have a pair of hydrogen bonds. When you have that one is guanine and cytosine, you have a 3 bonds. Okay, and so on and so forth. All right. So same thing. So, and if you substitute thymine with uracil, okay, if you recall, the only differences between a thymine and a uracil is that metal group over here. So you can still form 2 bonds with an adenine. Okay, so connectivity in biology, A pair with T, C pairs with G. In RNA, it's just that one is being replaced. Okay, but the funny thing is a recent study found a 6 nucleic acid in nature. So we will see where we go from here. Maybe we will mutate into an alien. Maybe not. I'm not sure, but we will see. Maybe not my son, because my son is already a normal human, but maybe your sons and daughters. I'm not sure, but yeah. Okay. This is another. This is basically what I've just said in short. Okay. If you want to look through, then you can just zoom in into the lecture slide. And what is missing is what are the relationship between RNA and amino acid. Okay. So we will go into this section in about 5 minutes. And if we are not managed to, if we are unable to complete this today, we will continue next Tuesday. Okay. So protein biosynthesis. So this is where the RNA is linked to a protein or a peptide. So before that, let's learn biology. Yay! Yay! I put it there. Okay. So this is a cell. This is the basic structure of a cell. That is what we call as the nucleus, the central, the brain of a whole cell. So it contains a membrane, the lining outside of the nucleus. You have a nuclear pores. So in the middle here, there's small pores that allows for the RNA from DNA, because you know that RNA is a duplication from a DNA. So the RNA needs to move out from the nucleus for it to synthesize protein. So this pause allows RNA to move out, but not DNA. So DNA stays in, RNA moves out. Okay. So it has a nucleolus, which is the yellow one over there. Okay, I'm not going to talk about it. And basically, it is where we host our genetic material. So each individual cells have a nucleus except for our red blood cell. Okay, so red blood cells lost its nucleus as it grows up. So in the beginning, when it was a baby, we have a nucleus. As it grows and grows and grows, the nucleus being taken out. So that's why if you look at red blood cells from the side, if you remember in high school, your teacher will always draw it like that. Okay, so that in the middle there is not because the red blood cells are on diet. It's because originally the nucleus is there, but when you take out the nucleus, there are now more space inside the cell. Thus, it kind of like becoming a thin type, a thin layer, not thin layer chromatography, a thin layer red blood cells. Now, other than that, what else do we have? We have the cell membrane, which is on the outside. We have the cytoplasm, which is the matrix, or the matrix, the liquid component of the whole cell. Okay, you have endoplasmic reticulum, which is the one next to a nucleus. So an ER complex will always link to a nucleus. So this is where your ribosomes are normally in. Okay, and this is where normally the protein synthesis takes place. So it's very easy. DNA will move to RNA, and then RNA moves out, and then straight after it encounters a ribosome, and then ribosome produces protein. So these are all the basic idea. Of course, it can be more complex than that, especially if a virus comes out from the outside. How does it transfer the RNA and so on and so forth? We're not going to look at that. Okay, so this is just a basic biology. And then the green one is a Golgi complex. This is where when you have a protein synthesized, the protein might not fall on its correct conformation, thus it will not function properly. So this is where the processes to fold, well, one of the processes, of course, whereby the post-translational modification takes place. Okay. Oh, I do have two Golgi apparatus. And then we have ribosomes inside there. Ribosome can also be in the cytoplasm, okay, but mostly are in the ER complex. And finally, we have our powerhouse, the mitochondria. So this is where we get most of our energy. So if you are too energetic, probably you have too many mitochondria. I'm just joking. Okay, so it doesn't work that way. So mitochondria is just a powerhouse for your body. So if you recall in high school, you might learn about aerobic and anaerobic respiration. Okay, so the principle takes place in a mitochondria. Okay, we have 3 more minutes. So to sum it up, of course, I will not go into very detail, because if it's very detailed, then there are so many proteins involved. I'm just going to go very brief, basic idea. You have your DNA over here, okay, it being transcribed into mRNA, okay, and from mRNA over here, it being translated by the ribosome into a polypeptide or protein. Okay, so these are the two terminologies in protein biosynthesis. Other than that, what else are involved. So you have this RNA polymerase. Okay, so what RNA polymerase does is it unwind the helix, the quadruple helix, no quadruple, the double helix nature of DNA. Okay, so it opens up the DNA like that, and then it starts to do a duplication. Okay, it copies the genetic sequence as it moves along the DNA. Okay, you might ask me, and then how did it falls? How did it form a double helix again? Well, there's actually another protein that is involved in the process and whatnot. But yeah, I'm just not gonna go into very detail about DNA replication. Yeah, unless if you're interested, then you can just find the information, and you can always consult with me what actually happened if you don't understand about the processes. Okay, but this is how the protein synthesis works. Okay, you have DNA forming an mRNA, and then mRNA is being read by ribosome, and ribosome produces the polypeptide. Of course, what is not shown here is the tRNA bringing in the amino acid. Okay, so tRNA has a sequence that can be complementary to the mRNA sequence. At the same time, it carries an amino acid. Okay, so this is how protein can be made very, very quickly, very, very specifically, because it is very specific, as we will see in the next slide. Okay, so two processes, as I mentioned, processes involve two main enzymes. One is RNA polymerase and one is a ribosome. Of course, I put it there too because for the sake of our course, I will just mention two, but in reality, there are more than two. At least there's, if I'm not mistaken, it's about five or that was discovered that is involved in either DNA replication or mRNA production. All right, so this is how RNA actually helps in producing a protein. Okay, if you see on the right hand side, on the table right hand side over here, so each individual 3-letter code of an RNA will correspond to one amino acid. Okay, so for example, again, why is it U? Because it is an mRNA sequence. Okay, so if the sequence is UUU, then the ribosome will translate UUU into a phenylalanine. So you have your ribosome. I think we have a ribosome over here. Okay, for example, a ribosome over here. Okay, and say for example, it says here AUG. Okay, so AUG is a start codon. What does it mean? It means that all protein will start with the same sequence. So all protein will have methionine as the first amino acid. Okay, and then as you go, the first one is methionine, AUG. If you look at the table over here, you go A, and then U, and then G. G is AUG over here. Okay, sorry, AUG is over here. That one is a column for U. Okay, this is a column for A. Okay, so A, first row, U, and then G. So AUG always code for methionine, and as you can see here, methionine has only one code. AUG, as compared to everything else, has two codes. Okay, so it is always the first start of every single protein in our body. As simple as that. Okay, so that's why we call it as a start codon. Then how do you read the codon? So codon is this 3-letter code. Okay, we call it as a codon. So to know what is the next amino acid is you need to move to a 3-letter code on the next one. So the next one is CCG. Okay, so if you look at C and then another C and then another G, proline. So you know that the second amino acid for the peptide will be proline. And then the next one is UAU. So you go with U and then A and U, you know, it's tyrosine. So next one will be tyrosine. The next one will be and next and next and next. Okay, so that's how protein synthesis works. So the 3-letter code gives out a very, very high specificity. That's why our body can produce multiple copies of proteins in very, very sensitive manner, and every single protein will definitely function in a similar manner. Okay, so this is one of the reasons. And of course, it sounded so weird. Why does every protein start with methionine? Well, methionine is just being used as its first amino acid during the synthesis. However, during post-translational modification in the Golgi apparatus, okay, this will be cleaved off. Well, of course, it depends on the protein itself. So some protein might have it, some protein might not have it. So it all depends on the biological process, and it will be cleaved off. Okay, so if you recall, if you look at the amino acid sequence of an insulin, it doesn't start with methionine at the N terminal. So this is at the N terminal because it is the first amino acid, and a biosynthesis starts, as I mentioned, from N to C. While when we are doing a synthesis, for example, using a solid phase peptide synthesis, we will do from C to N. Okay, so this one is synthetic, this one is biosynthetic. All right, so I think that is all for today. A quick practice, you can just have a practice, have a go, and we will continue on next Tuesday. All right, so thank you, and take care, stay safe, everybody.
Thank you, doctor. Stay safe.
Thank you,
Thank
doctor.
you, doctor.
Doctor.
Take care, doctor.
Thank you, doctor.
Thank you,
Doctor.
doctor.
Thank you, doctor.
Thank
Bye,
you, doctor.
doctor.
Bye.